Chief of Research, Division of Geriatrics, Gerontology, and Palliative Care at the Department of Medicine, University of California at San Diego. Dr. Molina's lab focuses on using mitochondria as a proxy to study human aging process. Although the importance of mitochondrial function in both healthy physiology and disease pathology and its relation to aging and many age-related disorders is widely recognized, the specific details by which the two are mechanistically linked remain unclear. Dr. Molina's lab therefore takes a deep dive into mitochondrial function and characterizing cellular bioenergetics across various ages, conditions, and treatments. Additional research, which is conducted at the Molina lab, include identifying antecedent biomarkers and risk factors that predict later life Alzheimer's disease vulnerability or resilience, and finding circulating factors that mediate systemic age-related bioenergetic decline. And finally, another central goal of the Molina lab is to understand the mechanisms underlying the decline of physical function with advancing age and age-related conditions such as heart failure and obesity. It is our pleasure to have Dr. Molina with us today, and we look forward to hearing from you. A small housekeeping note, please, as always, put in your questions in the Q&A box. Thank you very much. Welcome, Dr. Molina.
All right, good morning, everyone. Can you hear me okay? Awesome. Thank you, Sai, for that introduction. And good morning to everyone. Happy Valentine's Day. Thank you for choosing to spend this time with me. And thank you, of course, for the opportunity to come and present this morning. I'm a big fan of Boston, lived there for almost a decade, and wish I could be there in person. So let's go ahead and get started. First, I'd like to get my disclosures out of the way. Most of the work I'm going to be presenting on today is supported by the National Institute on Aging and also a grant from the American Heart Association. Our lab's interest in mitochondrial bioenergetics and healthy aging is based on this three-part premise. First part of this premise is that one's genetics and chronological age are what determines that individual's baseline mitochondrial function over time. And that's represented by this dashed line, which of course has a downward trajectory, with age-related bioenergetic decline being one of the most well-established hallmarks of the aging process. We then think about behavior, things like nutrition and lifestyle, and also one's environment as things that can determine the rate or trajectory of these bioenergetic changes over time, as represented by these red and green lines. Now, where you fall in this graph, or your resulting bioenergetic status, we believe then influences your risk and progression for age-related diseases and conditions. So we think about mitochondrial bioenergetics, or the measurement of mitochondrial function, as an integrative and cumulative and measurable outcome that can report on biological age, but interestingly is one that's also modifiable by interventions. So one of the central questions in my lab is whether we can use human bioenergetic profiling to advance precision healthcare for older adults. So over the past decade now, we've been engaged in a number of areas of interest, some of which are listed here. We're interested in patient safety, whether we can predict whether a patient can safely undergo and of course benefit from a procedure or intervention. We're interested in improving outcomes, figuring out whether treatment can be personalized based on that person's unique bioenergetic capacity. We have a big program in disease prevention, as I mentioned, recognizing antecedent bioenergetic biomarkers so that preventative strategies can be implemented. And then we're also interested in monitoring whether or not we can use bioenergetic profile to track disease progression or response to intervention. So when we think about human bioenergetic profiling, well, there's a number of current approaches. And these include invasive biopsy-based approaches, minimally invasive blood-based approaches, and also non-invasive approaches. These biopsy-based approaches are focused on detailed analysis of the tissue of interest. And that's why this still remains the gold standard assessment of human bioenergetic capacity. A lot of publications have been looking at skeletal muscle fibers, of course, and mitochondrial function in that tissue. In my lab, we do a lot of work doing blood-based bioenergetic profiling, which also allows for detailed analysis of mitochondrial function, but uses circulating cells as surrogates that can report on systemic bioenergetic capacity. And I'll talk a bit more about that in a second. One of the things that we like about the blood-based approach is that samples can be collected at multiple sites and sent to a central facility, such as our lab, for analysis. Of course, there's non-invasive approaches. I'm not going to talk about these today. This is not our area of expertise. These are imaging-based approaches that can report on tissue metabolism. So as I mentioned, my lab really specializes in mitochondrial respirometry to examine the electron transport system. So this approach capitalizes on the activity of complex 4. So you may remember that as electrons are passed down through the electron transport chain, when the electrons arrive at complex 4, they bind with molecular oxygen to form water. And it turns out that by measuring this consumption of oxygen at complex 4, we can get a readout of the current of electrons that's passing through the electron transport chain. And then we can then measure this current as we add various fuels, substrates, and inhibitors. So for example, we can add fuels and substrates for fatty acid oxidation, and then layer on additional fuels and substrates for complex 1, complex 2, and glycerol phosphate. So giving us the maximum capacity for oxidative phosphorylation. We can then uncouple the system and also measure maximal respiration of mitochondria. So we've been utilizing this approach in various clinical studies. Currently, our studies are largely focused on Alzheimer's disease, heart failure, and also physical ability and frailty. So what I'd like to do this morning is to take you through some examples that I hope I've chosen well and are of interest to you. And the first example I'd like to talk about is our work on heart failure with preserved ejection fraction. So HFpEF, as this is commonly referred to, is a disease of aging. It's the most common cause for hospitalization among older adults. It now accounts for almost 50% of all heart failure cases. And this number is predicted to continue to increase because of the common risk factors for HFpEF include things like obesity, diabetes, and older age, things that are all becoming more prominent in our population. Interestingly, the primary manifestation of HFpEF is severe exercise intolerance. And as I'll hope to show you, HFpEF has several features of accelerated aging. So far, therapeutic targeting of cardiac function for HFpEF has largely failed. However, the treatments that have shown promise include things like strength training and caloric restriction, interventions that we know will have systemic effects. So interestingly, there's also multiple lines of evidence indicating that it's actually impaired skeletal muscle metabolism that's underlying exercise intolerance in these patients. Again, that's the primary clinical manifestation of disease. So one of the first questions we wanted to ask was whether mitochondrial bioenergetics was playing a role here. So we did a cross-sectional comparison looking at patients with HFpEF and healthy controls. You see that they're pretty well matched for age and gender distribution, but that things like weight, BMI, fat mass, and percent body fat are all much higher in the patients with HFpEF. Again, this is a characteristic of the disease itself. Now, HFpEF exercise intolerance, well, we certainly saw that in our cohort with much reduced peak VO2 in our HFpEF participants, and also much reduced 6-minute walk distance. Now, what we did was to perform high-resolution respirometry of freshly biopsied permeabilized muscle fiber bundles from these participants. Here's the respirometry protocol we used in case you're interested in seeing that. But what we found was that across the board, all of these bioenergetic parameters were lower in the skeletal muscle of HFpEF patients versus the healthy controls. Now, let's look specifically at max ETS. This is our maximal capacity. And you can see that there's really good separation between the max ETS of our HFpEF patients and the healthy controls. And then with both groups together, we see a significant positive correlation with exercise capacity as measured by peak VO2. Again, we see a really strong separation between our two groups. We also looked at physical function as measured by the short physical performance battery. And we do see that separation between our two groups. However, as expected, you do have these ceiling effects with the SPPV only going up to 12. But when we look at individual components of the SPPV, such as 4-meter gait and chair stand, again, we see the expected, in this case, negative correlations with max ETS. And again, the strong separation between the HFpEF patients and the healthy controls. So as I was saying earlier, our lab has really made a lot of headway in this area of blood-based bioenergetic profiling. And this approach is based on the hypothesis that measures of mitochondrial function can be performed in circulating cells to integrate the effects of environment, genetics, nutrition, lifestyle, things I mentioned previously, on systemic bioenergetic capacity. Now, as opposed to the tissue-based approach that I just showed you with HFpEF, certainly the blood-based approach has a number of advantages. It's minimally invasive. It's suitable for a wide array of patient populations. You can do serial assessments to track mitochondrial function. And again, it's suitable for multi-site or off-site clinical studies. The premise for blood-based bioenergetic profiling is pretty simple. The premise is that blood cells are going to be continuously exposed to circulating factors that we know mediate mitochondrial function across multiple tissues and organs. Now, if this premise is correct, then blood cell respirometry should be able to recapitulate the bioenergetic capacity of other tissues. In particular, highly metabolically active tissues such as the brain, cardiac muscle, and skeletal muscle. So in order to test this premise, we wanted to see if blood cells could mirror the skeletal muscle bioenergetic capacity. So this was a study published a few years ago now, where we looked at skeletal muscle fiber bundles from non-human primates and did contemporaneous respirometric profiling of monocytes. And again, we see this significant positive correlation with both complex 1 and 2 mediated skeletal muscle fiber respiration and complex 1 mediated respiration. Similarly, we saw correlations with cardiac muscle. In this case, this was isolated mitochondria, and we saw a significant positive correlation, specifically with platelet maximal respiration. Now, we think that these results may underlie some earlier studies that we published showing that blood-based bioenergetic profiling was also related to the physical abilities of older adults. So we previously published that the spare respiratory capacity of peripheral blood mononuclear cells is significantly positively correlated to gait speed among older adults, and also muscle strength, SPPV, muscle quality. Now, we also looked at potential correlations with the brain, and we saw similar findings. So here we are comparing monocyte maximal respiration to maximal coupled and uncoupled respiration of mitochondria isolated from the frontal cortex. Now, again, here we see a positive correlation again, but so far everything I've been showing you is kind of comparing respirometry to respirometry. So we also had an opportunity to take these non-human primates in for FDG PET CT imaging to look at brain glucose utilization in the frontal cortex, amygdala, and hippocampus. These are the 3 places we looked at. And we saw that across these 3 brain regions, the measures were significantly positively correlated to monocyte bioenergetics. Now, we think this correlation to the brain is particularly interesting when you think about the energy demand of the brain. So while the brain only accounts for about 2% of your total body weight, it also consumes 20% of your total body oxygen at rest, and it represents 60% of total body glucose utilization. That's like 420 calories going to your brain at rest each day. However, because of this very high metabolic demand, we think that underlies the reason why the brain is also susceptible to the effects of systemic bioenergetic decline, such as what you have with aging. So a postdoc in my lab a couple of years ago wanted to see if bioenergetic capacity, measured again in blood cells, could be related to human brain architecture. And to answer this question, she took advantage of an ongoing study called African American VHS Mind. This study is a cross-sectional genetic and epi study that was looking at risk factors for impaired cognitive performance in African Americans with type 2 diabetes. What she did was to bring back 16 participants, and this is meant to be a quite diverse cohort. So looking at ages from 50 to over 80 years old, BMIs from 25 to 50, and different durations of type 2 diabetes. Again, the goal here was to get a wide dynamic range within this relatively small cohort. And what Gargi, Dr. Mahapatra found was that PBMC bioenergetics was related to white matter volume and total intracranial volume. So here we're looking at basal respiration and maximal respiration, but then when we look at individual components, we see that almost across the board, we see these significant positive correlations. And interestingly, the fatty acid oxidation mediated respiration in PBMCs was particularly strongly correlated to both white matter volume and total intracranial volume. So this really paved the way to our growing interest in Alzheimer's disease. So as many of you know, there are still no preventative strategies or therapeutic interventions that are really proven to be effective for AD dementia. And this is likely because AD pathology, including the developments of plaques and tangles, is occurring years before the manifestation of clinical symptoms. So any efforts to prevent or countermand AD dementia are going to rely on early detection of pre-symptomatic pathological changes. And among these, mitochondrial dysfunction has been proposed to be a primary event leading to some of the hallmark brain pathologies in AD. So our goal for this study is to identify antecedent bioenergetic signatures that predict later life AD vulnerability or resilience. And to do this, we're examining transitions from normal aging to MCI and from MCI to AD and related disorders. Of course, to get this grant funded, we had to have some preliminary data. And this was one of the pieces that really showed to us that we were onto something. So here, we're looking at PBMC maximal respiration and basal respiration and comparing it to the presence of A-beta 42 in the CSF. So just as a reminder, if A-beta 42 is in the CSF, that's likely a good thing, because it suggests that it's getting cleared from the brain. And indeed, we saw that with higher blood cell PBMC respiration, we saw higher levels of A-beta 42 in the CSF. So we have now embarked on a longitudinal clinical cohort study that's utilizing blood-based bioenergetic profiling to look at AD risk and progression. This is a cohort study that's being done with the Wake Forest ADRC. We're looking at 500 older adults with different degrees of AD risk and pathology, comparing cognitively normal, MCI, and AD. We're actually currently in year 4 of the study. But at years 1 and year 4, we're doing complete blood-based bioenergetic profiling. There are yearly cognitive assessments and multiple other readouts of AD pathology. What I'd like to show you this morning is our analysis or interim analysis of the baseline data. So this is a quick description of the cohort that we have so far. So we do see decreases in the Montreal Cognitive Assessment score, starting with the participants with MCI. 3 MMSE scores were lower, significantly lower in the participants with dementia. And when we looked at MPAC-5, this is a cognitive Z-score looking at different cognitive abilities, we see the stepwise decline as we go from normal cognitive MCI to dementia. This dementia we call dementia due to probable AD, because these participants have lower cortical thickness and changes in white matter hyperintensities. Now, when we looked at PBMC bioenergetics, in this case, just by high throughput respirometry, we see significantly lower basal, maximal, and spare respiratory capacity in the participants with dementia. However, when we did our high resolution respirometry, looking at maximal ETS, we see a stepwise decline as we go from normal cognitive to MCI to dementia. So I want to change the topic a little bit, because so far what I've been showing you is that lower bioenergetic capacity is associated with worse things. However, I still don't think we fully understand what an optimal bioenergetic profile is. I think optimal to me suggests, matching metabolic demand with energy production. So one of the ways we're starting to better define what an optimal bioenergetic profile is to look at, behaviors and lifestyles that we know are good for you. So among the diets that we know are good for you is the Mediterranean diet. This diet is, as you all know, is associated with improvements in insulin sensitivity, lipid profile, blood pressure, and inflammation. Then if we think of the converse of this in an unhealthy diet, well, a Western diet certainly comes to mind. This is a diet that's associated with obesity, cardiovascular disease, hypertension, diabetes, and high cholesterol. So in collaboration with the Primate Center at Wake Forest, we did a non-human primate study to look at the effects of these whole dietary patterns. So animals were given Western or Mediterranean diets for 30 months. This is the equivalent of 8 human years. Animals on the Western diet ate things like lard, beef tallow, high fructose corn syrup, whereas the animals on the Mediterranean diet received things like fish oil, olive oil, and fruit puree. Now, the interesting thing about these diets was that the major macronutrients were pretty well matched. So same percentage of calories coming from protein, carbohydrates, and fat, but what was different was the types of fats that we were providing. So with the Western diet, more saturated fat, with the Mediterranean diet, more monounsaturated fat. And again, other things as well, looking at more fiber in the Mediterranean diet and lower salt in the Mediterranean diet animals. Now, at the end of the 30 months, we performed, again, highly detailed, high resolution respirometric profiling of permeabilized muscle fiber bundles from these animals. And again, here is the protocol that we used. And what we found, interestingly, across the board was that the animals on the Western diet had higher skeletal muscle mitochondrial respiration compared to the animals on the Western diet. And as you can see, we looked at a lot of respirometric parameters. Focusing in on max ETS, again, this is the max capacity, we saw that these higher rates of respiration were also associated with insulin resistance. So here's a case where higher respiration is associated with worse outcomes. Now, we started to, we were starting to look at whether or not these same sort of patterns held up in the human population. And we're starting to see signals from a study based out of Cilento showing us the same sort of thing, that participants from Cilento had relatively lower bioenergetic capacity compared to participants in Cilento, in San Diego. However, this is a study that's still ongoing. These are actually pictures from the last international trip I took before the pandemic struck. So we're hoping to get back to the study and to see if these sorts of findings hold up in the human population. So to be continued on our study of the Mediterranean diet. But what we are starting to think is that with the Western diet, skeletal muscle mitochondria are potentially overstimulated and therefore potentially more susceptible to damage with advancing age. Now, we also looked at the brain in the non-human primate study. And there what we found was also quite interesting. We didn't see any profound differences based on which diet the animals were given. However, what we found was that with the Mediterranean diet, if we looked at mitochondria isolated from different brain regions, in this case, prefrontal cortex, entorhinal cortex, and cerebellum, there's this pattern of higher respiration in the prefrontal cortex and lower respiration in the cerebellum. However, when we did the same sort of analysis with brains from animals on the Western diet, you see that these patterns are scrambled. And also, we find that with the prefrontal cortex and entorhinal cortex specifically, you start seeing correlations with fasting blood glucose. So this is potentially interesting based on the growing recognition that there's a link between prediabetes and risk for AD. Because the prefrontal cortex and entorhinal cortex are the regions where you would expect to see AD pathology the earliest, whereas the cerebellum is relatively protected from AD pathology. All right, so far I've been showing you that there seems to be the systemic nature to bioenergetic capacity. And when we compare different organs to the blood, we're seeing these patterns emerge. Now, this started to get us to think, whether or not we could figure out what the drivers of systemic bioenergetic capacity were. And one of the things that we decided to look into was whether or not circulating factors could be mediating why these different organs and blood cells were correlating to one another. And we do know from previous studies that circulating factors mediate key aspects of aging. And the best, I think, evidence for that so far has come from parabiosis studies. So parabiosis studies showed that heterochronic parabionts, this is when you connect the circulatory systems of young and old mice surgically. We find, or others have reported that there are systemic rejuvenating effects for the older parabiont, whereas there are deleterious effects for the younger parabiont. And this has been reported in multiple tissues, including the CNS, skeletal muscle, liver, heart, and pancreas. So what a grad student in my lab wanted to look into was whether or not circulating factors could mediate age-related bioenergetic decline. So she connected the circulatory systems of young and old mice for 7 to 8 weeks. She also had isochronic young controls and old controls. At the end of the 7 to 8 weeks, she collected soleus muscle, permeabilized the skeletal muscle fiber bundles, and performed higher resolution respirometry, a relatively simple protocol here. And here is what she found. So let's just focus on max ETS again. It's really the same pattern, no matter which respirometric parameter we look at. But first of all, as expected, if we look at the young animals from the young isochronic controls, they have the highest rates of max ETS. And when we look at the old animals from the old controls, they have the lowest max ETS. Now, interestingly, if we look at the young animal from the young-old heterochronic parabiosis pair and the old animal from the young-old parabiosis pair, they look more like the old controls. And strikingly, they start to equalize their bioenergetic capacity. So this shows us that circulating factors, in this case, are primary mediators of this age-related bioenergetic decline. Now, this was true not just for mitochondrial respirometry. We actually saw a similar pattern when we looked at changes in mitochondrial morphology. Here, the effects were even more striking. Again, largest mitochondrial area and average mitochondrial area in the young controls. Old controls had the lowest, and then the two heterochronic animals looked a lot like the old controls. So again, circulating factors mediating the negative bioenergetic effects of aging in this case. All right, so I want to go back to our central goal of advancing precision medicine, because if we are able to identify the circulating factors mediating age-related bioenergetic decline, where clearly this is going to have very important diagnostic, prognostic, and therapeutic implications. So the problem here is that this is going to be a really complicated set of experiments. So if we think of the potential mitoactive factors in whole serum, well, there's a whole list of possibilities. There's microbiome-derived factors, various proteins, metabolites, vesicles, and also inflammatory cytokines. So what we're going to need is a better discovery-based approach for identifying what these individual factors are. So this is something we've been working on for a couple of years now. And what I'd like to give you today is an example of how we're utilizing this discovery-based approach in an exercise and weight loss study. So what we did was to go back to a previously completed study that's called INFIT, which starts from Improving Muscle for Functional Independence trial. The PI of this was Barb Nicholas at Wake Forest. And what her group did was to enroll overweight to obese older adults and put them on 5 months of resistance training 3 times a week, with and without caloric restriction. Now, this caloric restriction was a 600 kilocalorie deficit based on estimated daily energy needs. At the end of the 5 months, the average weight loss was 5.7%. Resistance training did improve muscle strength and physical function, and adding caloric restriction did result in a loss of lean mass, but didn't compromise the functional benefits of resistance training. Now, interesting to us, this response to intervention was highly heterogeneous. So not everyone benefited from the exercise intervention. In fact, there is a good proportion of participants who actually had worse knee extensor strength and worse usual gait speed after the intervention. So we began to ask whether the response to intervention could be mediated by mitochondrial bioenergetics, and if so, whether mitoactive circulating factors are involved. So to address this question, our first step was to isolate the bioenergetic effects of circulating factors. And the way we did this was to seed naive cells onto a plate. After an overnight incubation, we changed the media to media containing dilute serum from INFIT participants. This is serum collected before and after intervention. And then after that, after 24 hours, we then did seahorse respirometry paired with automated plate-based imaging. So what we could do was then take these bioenergetic profiles and show the data normalized to cell count. And here's what we found. So first, let's look at the resistance training intervention alone. What we found is that cells that were exposed to post-intervention serum had higher bioenergetic capacity, again, just looking at max here, compared to the same cells exposed to pre-intervention serum. So this is showing us that serum alone contains factors that can mediate bioenergetic improvements associated with, in this case, strength training. Now, with the addition of caloric restriction, we still see an improvement comparing pre and post. However, the effect is somewhat blunted. It was only statistically significant with maximal respiration. Interestingly, the serum-mediated bioenergetic effects were also heterogeneous, similar to what we saw with the effects of the effects on physical ability. And we had some negative responders and positive responders. When we looked at correlations between, in this case, MAT-SF, this is perceived physical function, the change in MAT-SF and the change in maximum respiration, as you compare the effects of serum pre and post intervention, there's a positive correlation, pretty mild correlation. And then same thing with the change in the 400-meter walk, not quite statistically significant, but again, pointing in the same direction that improvements in the serum-mediated bioenergetic capacity are associated with improvements in physical function. Now, to try to identify specific molecules associated with the exercise response, we were looking for those molecules that were, first of all, upregulated in the negative responders, And then secondly, negatively correlated with the in vitro bioenergetic effects of the serum sample. So again, we're reducing this down to just what the serum itself is doing in vitro. And here's two molecules that we found to meet these criteria. Again, this is a discovery-based approach, so I'm not even sure what these are. I'm sure some of you guys could hopefully tell me. But here are two molecules that were negatively associated with a change in maximal respiration. And then when these two molecules were commercially available, when we put them directly onto cells, again, in the C2, C12 cells, we saw that with increasing doses of these molecules, they had inhibitory effects on mitochondrial function. So again, still early days for this approach. Probably took us a couple of years just to get to this point. But what we're excited about is that this discovery-based approach can be applied to various studies to identify mediators of mitochondrial function. And we're happy to work with people with completed studies that have stored plasma or serum samples. We're interested in these sorts of questions. Also, the example I showed you was based on metabolomic analysis, but this can be combined with different omic platforms. Now, as we're moving more towards this kind of in vitro validation, we do recognize that we have a need for new research resources for in vitro studies, including the need for better cell models of aging. So what we'd like to get to is to a point where we can actually test our hit molecules on cells that are representative of specific cell types according to different human ages. So this is what we're calling cell models of aging. These are resources that we're currently working on, and we're hoping to make available in the upcoming year through our new San Diego Nathan Schock Center. This center was established in 2020, and the theme of our center is the heterogeneity of aging. Our Nathan Schock Center is a collaboration between the Salk Institute, which is the leading institution, our group at UCSD, and also the Sanford Burnham Previs. So what we've been doing over the past year now is enrolling a cohort of participants who are 20 to 90 plus years old, conducting extensive clinical and cellular phenotyping to characterize biological age, and then generating human cell models that represent the adult human life course. So these human cell models are going to include subject-specific fibroblasts, but then importantly, these then give us the opportunity to create induced cell models and human iPSC models. So why is this significant? Well, this is really based on seminal work by Dr. Rusty Gage, where he showed that fibroblasts can be directly induced to cell types of interest, in this case, neurons, and maintain age-related phenotypes. Whereas if you go the iPSC route, the reprogramming step actually rejuvenates the cells so that when you then have your cell type of interest, then these have a rejuvenated phenotype. So what we're able to do with these two types of cell models is to compare age-related versus non-age-related phenotypes using cell models derived from the same participants. So with that, I just want to acknowledge the people in my lab who have actually completed the work that I shared today. Jenny and Phil have actually moved on from the lab to faculty positions and industry positions. If you'd like to learn more about what we do, feel free to check out our website. We also have a student-run Instagram account if you'd like to know what we do when we're outside of the laboratory. And one final announcement, we are recruiting at UCSD. This year, we have 2 tenure-track faculty positions that we're recruiting for at the assistant and associate professor levels. The 2 areas of research that we're particularly interested in are geroscience and gerotechnology. If you'd like to learn more about these opportunities, please shoot me an email and let me know. So with that, I'm going to go ahead and stop my share and happy to open it up to questions.
Great. Thank you so much, Dr. Molina. That was a fascinating talk and we really learned a lot. So on to the questions coming from our attendees. The first question asks, do you know what is underlying your observed shifts in bioenergetic capacity? For example, is it due to increased mitochondrial biogenesis, mitophagy, or mtDNA levels? Lower mtDNA mutation load, maybe? Altered substrate utilization, that's another sort of question. Basically, the question asks, where is it coming from and does it change with the interventions you use?
Yeah, that's a fantastic question. And I think some of the answers, some of the examples I showed you today, the changes were so gross that across the board, everything we looked at was different. So if you remember some of the respirometric protocols I was showing, those were actually looking at specific contributions of different fuels, so fatty acids versus glucose, different entry points, such as complex 1 and complex 2. But again, like for example, with the parabiosis study, really the effects were across the board. So that could mean that it is simply a change in mitochondrial content. However, we just need to go back and actually do those assays to figure that out. And looking at the other things, so as far as biogenesis versus mitophagy versus mitochondrial DNA, so yeah, we are trying to look into some of those questions by looking at specific markers, and hopefully we'll get a handle on some of those soon. We'd love to know, how the effects differ based on the different interventions. Again, that's a future and ongoing work for us.
Thank you. And that first question was from Dr. Christopher Wiley. We have another question from Dr. Shangdang Wang. He asks, have you seen any gender differences on systemic bioenergetic capacity, example, respirometry from your studies?
Yeah, this is a great question and one that, remains open. So I was part of a publication that came out recently in FASIB Journal, where our group did a meta-analysis across multiple studies and, didn't see any sex differences across the studies that we looked at. And this wasn't just respirometry, there were other sorts of measures as well. However, we do have other studies where we've looked into this, in our cohorts where we are seeing sex differences. And interestingly, these sex differences seem to be more present at older ages versus younger ages. And we're actually working on a publication to share some of that data soon. So great question, open question, and hope to share more in a little bit.
Great. If others would like to ask questions, please go ahead and post them, or you're welcome to go ahead and ask if that's something that's open. No?
Awesome.
Roger?
Questions are coming in, Anthony. Thank you for an excellent presentation. I really enjoyed it. For people that don't sort of play in this area, could you explain a little bit about some of the different approaches to assessing mitochondrial energetics? The Ouroboros techniques, permeabilized muscles, seahorse, for example, and maybe just so people understand sort of what the limitations and strengths of those are.
Yeah, happy to do that. Certainly in my lab, we use both. We've been working with the Seahorse folks since they were, a small biotech company before the Agilent acquisition, and we have an ongoing partnership with Ouroboros Instruments. From my perspective, these systems are highly complementary to one another. So for those of you who aren't familiar, the Seahorse system is a plate-based system. The ones that we use have a 96-well format. We're able to pair our Seahorse system with a digital imaging system that allows us to then take our respirometry data and match it with imaging data to tell us about things like cell count. So it's a great high throughput system. We use it quite often for screening. So for screening, a bunch of molecules, or if we are screening through a set of serum samples for a study, we think of that as a great first step. And what it'll tell us is grossly, is there an effect on maximal bioenergetic capacity or basal respiration, or what they call spare respiratory capacity. Those assays are typically done with intact cells, but certainly, you can do other assays with the Seahorse system where you permeabilize and look at the activities of individual complexes. However, in our lab, when we want to get down to that level, we tend to use the Ouroboros O2K system. This is a chamber-based system, low throughput, only 2 chambers per machine. So we end up having to use multiple chambers for our studies. But the way we're able to do that is, the way we do those studies with the permeabilized cells is then kind of like what I was showing earlier, where we can put within an unlimited number of injections, different fuels, substrates, and inhibitors to probe the electron transport system in greater detail. And that's why we call it high resolution respirometry. Other considerations you might have are, whether you have a tissue. So tissues tend to do better in chambers where you don't have to stick them down to a plate and they can remain in suspension, or if you have cells that are not very adherent. So we go back and forth, really depending on the need for a particular study.
Great, thank you so much. We have a question from Dr. Sheldon Rowan. He asks, for a heart failure study, the cases have extraordinarily high BMIs, which can complicate interpretation due to secondary causes. If you match case and controls by BMI, are there still similar differences in mitochondrial function?
Yeah, it's a great question. And, there's really, clinically no way to tease apart, HFpEF and obesity, since, these do come together. The best we could do was to control for BMI in our analysis. And we find that even when controlling for BMI, the differences that we see still remain significant.
Great, thank you. I have one basic question, and that goes back to mitochondrial bioenergetics. Because of the relatively higher demands on glucose as well as overall energy by the brain, do you think that age-related mitochondrial bioenergetic decline would most significantly impact neurological and such diseases or disorders, or what's your thinking on that?
It's a good question. Certainly, if you look across neurological disorders, there does seem to be a role for mitochondrial bioenergetics, and that includes diseases of aging, but also diseases that are not related to aging. So you're right. I think because of the exquisitely high metabolic demand, this tissue does tend to be more susceptible. But we do see similar things when we look at skeletal muscle. So maybe pound for pound, it's not the same level of demand, but we sure have a lot more of it. And so I think that also underlies, why we're seeing such strong correlations with physical ability and physical function, even in, relatively smaller studies that we've done so far. I should say that, we are now involved in a much larger study that's focused on physical function and the development of mobility disability. This is a study called the Study of Muscle Metabolism and Aging. I think the first papers will be coming out soon, and there's a session at the IFCSR conference. But we're also finding, similar to that data I showed you with our AD cohort, similar strengths of correlations between skeletal muscle measures of mitochondrial function with physical performance measures such as gait speed and grip strength.
Great, thank you so much. And along those lines, I have one other question with regards to medication. Of all the major genre of medications that are, consumed by our older adults, and including maybe not specifically by older adults, antibiotics, do you see any specific, in your experience, class of medications that, contribute to greater or more accelerated decline in mitochondrial bioenergetics?
That's a great question as well. And to be honest, we haven't looked into it gotten into that level of detail in our studies yet. I know there are certain classes of medications where there are ongoing studies to look at effects on mitochondrial function. So, for example, there's a growing interest in the role on the effects of statins on mitochondrial function. So far, there's been a publication showing that the similar sorts of measures that we do with circulating cells do tend to be altered by statin use, and we're hoping to probe into that in the future as well in collaboration with the preventable trial. And then there is some interest in the effects of metformin on mitochondrial function. So we think of metformin not just as a complex 1 inhibitor, but also as a compound that could have long-term beneficial effects on mitochondrial function. There seems to be some evidence of hormetic effects. So that's, again, something that we have some data on, and we're still trying to piece together and publish.
Great, thank you so much. I don't see other questions posted. This was a very clear presentation and has given us a lot of, information for thought and further processing. But most importantly, we also look forward to all the work that you're doing, which is absolutely fascinating, and the intersection of interventions and the effects. We really look forward to all the findings and potential collaborations even maybe. And thank you so much for your time and wonderful presentation, and look forward.
Sure, thanks for having me. And yeah, if people are interested in collaborations, whether you have stored samples that you're interested in having us look at, or new studies that you'd like to collaborate on, I mean, we're here, and we're always happy to work together. Thank you.
All right, thank you so much.
Thank you, Anthony.